Good afternoon, everybody, and welcome to the second Retina Australia webinar for 2022, and a research update from Janssen. My name is Sally Turnbull. I'm the Administrative Officer for Retina Australia, and I will be the facilitator of this webinar today. As a participant, your camera and microphone will be switched off throughout. There will be an opportunity for you to ask questions once our speakers have finished their presentations. If you have any queries during the proceedings, you can type a question to me in the Q&A section. So Retina Australia is delighted to have the support of Janssen, who have recently provided generous funding for the redevelopment of the Retina Australia website, which will go live soon, as well as the introduction of Vega, an exciting new database system that has transformed the way we are able to interact with our members. This webinar provides an opportunity for you to hear about their work in research, testing, and treatment efforts for those living with inherited retinal disease. Our first speaker this afternoon is Felicity Sawford, Patient Engagement Manager with Janssen. Felicity holds a Bachelor of Science in Psychology in Biological Science from the Northern Territory University. She has so far a 22-year career in the pharma healthcare sector with experience in gastroenterology, cardiology, respiratory medicine, neuroscience, endocrinology, and more recently, pulmonary arterial hypertension, PAH. Felicity has spent the past 15 years working in partnership with healthcare practitioners, patient advocacy groups, patients and professional societies to improve speed of diagnosis, access to treatment, education, and to build communities in areas of high unmet medical need. She is currently the Patient Engagement Manager at Janssen for PAH and rare disease, oncology, and ophthalmology portfolios. So I'd just like to welcome Felicity, and we'll get started. Thank you so much.
Thank you, Sally, for the kind introduction. And I also need to thank Leighton and the rest of the executive at Retina Australia, both for your early and existing partnership with Janssen, but also your ingenuity in having myself and Marija come and speak to you today. We're absolutely thrilled to be here. And it's a wonderful opportunity for us to share our commitment that every day we're daring to envision a brighter future. Together, Marija and I will cover the topics of the importance of people engaging in their healthcare and the role of genetic testing and potential future treatments for inherited retinal diseases. Our presentations will cover three primary areas that we at Janssen are committed to working with you to achieve lasting change and healthcare outcomes in Australia. These three areas are engagement, how we will work systematically across multiple treatment areas to understand the experience of people living with complex healthcare needs and the opportunities of how this could work in retinal disease. The second is innovation, both from a scientific and a pipeline perspective, but also how we innovate to create solutions for the moments that matter most to you. And lastly, the opportunity that the evolving science in genetic diagnosis for research, treatment development, and access to future therapies may provide. Before I dive too deeply, I'd like to share a little of my world and my experience, and thank you, Sally, for reading that out so beautifully. I've spent the past 15 years working with patients, patient advocacy groups, doctors, nurses, and professional societies to raise awareness, improve time to diagnosis and diagnostic rigor, as well as access to treatments in the rare and life-threatening condition pulmonary arterial hypertension. And I'm really pleased to say that we have driven such considerable change, generational change within that period of time. So it's with considerable excitement that I bring those skills to learn a new area, which I hope very much that you will teach me more about. From a vision perspective, I'm also a sometime carer of my father who lives with a complex and chronic retinal condition. So the importance of caring is something I'm well familiar with. To share a small story, around the time of my eldest daughter's birth, and she's now 10, Dad was going through a series of diagnostic tests, surgical procedures, hospital stays, and some lengthy rehabilitation. And I'm fairly confident that Annabelle, at the tender age of 3 months, became the mascot of many hospital waiting rooms and doctor's surgeries where we spent hours, days, sometimes feeling like months. And thankfully, at least back then, she was quite well behaved. So I bring a carer's point of view and some understanding of vision and a deep history of patient engagement and shaping healthcare change. And I'm so excited to bring that together with Janssen and all of the, shall we say, the capabilities of the science and the amazing people that I get to work with, and now you, to work together towards a patient-inspired innovation. And that falls into three main categories: that we partner directly and we embed your voice into everything that we do, that we deliver therapies that best meet needs of people who are living with complex healthcare needs, and we do that as fast as possible with the overall aim to improving everybody's healthcare outcomes. So to do that, we have a deep commitment to develop medicines and solutions that transform individual lives and solve those important unmet medical needs. We do that with a clear eye on quality and safety at every stage of production, manufacturing, access, and once things are in market. And importantly, we understand that this is an evolving landscape and we need to respond and adapt to patient needs in meaningful ways at each stage. So we do this with transparency, and we do this with ongoing commitment that maximizes our best outcomes. And why do we do this? Janssen is the pharmaceutical companies of Johnson & Johnson, one of the largest healthcare companies in the world. And within Johnson & Johnson, patients have always been at the centre of everything that we do. It's in our credo, it's in our culture. Our credo was first written in 1943 and has served as the guiding principles for who we are and what we do across the decades. It's been updated from time to time to ensure we continue to meet the needs of today's world. But most importantly, and one very particular positive change in the world that we've seen in the last few years, at least, is that the role of the patient is changing rapidly. And today we're operating in an environment where patients are increasingly involved, educated, and engaged in their own healthcare. Our presence and your presence here today is a brilliant example of just that. So in response to this movement and this need of our current world, we're redefining our relationship through the creation of an entirely new patient engagement model, one where we interact directly with you, with your caregivers. We systematically and directly understand your point of view. We act early on those perspectives, right from drug discovery through to access to treatment. And we maintain this ongoing dialogue so that the solutions that we develop at each part of the journey meet your needs in the best possible way. To give you two key examples of how this has already happened, in particular, access to clinical trials can sometimes be quite difficult in markets where they are available. So we asked people, what are some of the barriers to participation? And importantly, we were told that transportation to hospitals and trial sites was a barrier. So to answer this need, we provided transportation. Seems very simple, doesn't it? Cutting the length of the visits in half and providing informed consent for people on paper or on a monitor to reduce glare where they had visual impairment. So those three things together, getting people to the trial sites, making those visits as efficient as possible, and giving people information in a way that they could access, meant that there was no dropouts within a 7-month period of the clinical trial. Now that's really important because the timely completion of clinical trials is a critical step towards understanding, is a medication going to work in the way it was designed to? And is it performing as expected? For another important medicine in development, we modified the target product profile, which is our guide for what a therapy needs to do. And that was based on patients' priorities. We were told that in this case, people find it quite difficult to commit to going onto a medicine if they have to wait a significant period of time to know that it will work. So in hearing those insights, coming to a solution as to how we could adapt to them, these are examples of early, systematic, and direct engagement, and everybody wins. So how do we go about this across the whole company? How do we make it bigger than just one patient engagement manager? Because this is something that we do as a whole of company. And that is, on the screen here, you'll see the 4 key principles, and that is that patient engagement becomes embedded in everything that we do, and that it is everyone's responsibility. Although you'll have someone like myself who'll be a key point of contact. That it's systematic. that we continue to check in across the entire life cycle of any therapeutic area in which we're involved. That we are accountable, that we measure our progress, that we check in, and that we promote a whole of industry change. And that we are dynamic. We can't see, if the last few years have taught us anything at all, it's that we need to be adaptable and we need to meet the needs of the day. And so identifying pain points and finding solutions and doing that continuously checking in is what we are committed to doing. To give you an example of how that looks across a life cycle of a therapy, everything from discovery, what are the unmet medical needs of our time? What therapies and what might work best in those areas? What are the formulations or delivery methods that are going to have the best outcome? And I can't wait for Marija to share with you some very, very funky science on how these things can happen. What clinical trial design is going to be appropriate? And what are the needs of the people who are involved in those trials? What do they report as being most important to them? Once we get through the clinical trial stage, then there's regulatory review. What are the policies? What are the diagnostic opportunities? And what are the access processes for any country in which we work? In this case, we're talking about Australia. And then once those therapies are available, how do we get feedback? How do we know where they're best working? And what are the experiences? And last but certainly not least, what are the educational materials and opportunities that people need and want at each point of their journey? Because what you need at the beginning when you're being diagnosed with something is not the same as what you need once you know a whole lot more about it and you're in the living with it stage. So to wrap this up for you, this is where your experience is super important, because we can see the intersections within a healthcare journey, but we're not walking in your shoes. And so we sum up patient experience, or we define it as the sum of all experiences a person has across their end-to-end disease journey, which provides a holistic view of what are the care needs and expectations. And we know that every patient is a person, and every person experiences their healthcare journey in a unique way. However, there are a number of things that happen for all of us. We spend time, and to give you an orientation on this particular graph, everything above the blue line in the middle is within a healthcare system, and anything below that healthcare line is self-care. It's when we're at home, we're at work, with our families, with our communities. And these things happen in a very similar way for almost every chronic healthcare condition. There's a period of prevention and wellness, a period of screening and diagnosis, a period of treatment decisions and interventions. Then we move into a monitor and manage phase, and then a living with the disease. And what you need to know at the screening and diagnosis phase is not necessarily what you will need to know once you have a diagnosis and you're on a course of treatment. So we have worked with a lot of different therapeutic areas up to this point to try and understand what are the moments that matter at each phase within this journey. And this is something I'm putting as a placeholder for a future discussion, because as we move into the areas of retinal diseases, we need to understand what does this journey look like for the people who are living it now and the people who will be living it in the future. And what are those moments that matter most? To do this with rigor and systematically, we need to have some tools in place to help us keep track of our progress. So patient insights and an integrated patient journey, as I've just shown you, this end-to-end experience that people have. We wrap this up into what's called a patient engagement index. And we can see across multiple different therapy areas. For example, what does it look like for the journey of somebody living with pulmonary arterial hypertension, for which I'm very familiar, to what are those similar needs for somebody who has another chronic healthcare condition, such as an inherited retinal disease? And where are those situations where your needs, whilst you have different conditions, are very similar at that point in your journey? I mentioned earlier about target product profiles. How do we make sure that we're meeting the needs of people at the appropriate point of time and giving them answers as soon as possible? That we value the customer voice, and that we have a way of incorporating those insights into things like formulation decisions, for example. How is a medication going to be delivered in the best possible way, both from an efficacy perspective, but also if it's something that, for example, not in this situation, but a medication you might be delivering to yourself For example, I worked in diabetes and people give themselves injections every day. How do we make that device the most user-friendly as possible? And that you have a voice in clinical trials and a voice to government when the time is most appropriate. And that we share best practice across all of these areas and we learn as we go. So lastly, as I said, working together towards a future where we have innovation inspired by patients and we hear your voice at every step. We partner directly with you. We deliver the therapies that you need when you need them as best as we possibly can to improve health outcomes. I'm leaving here again a few placeholders for how we might be doing these things in the future. And that's to do with both your clinicians, with patient support groups, such as the wonderful people at Retina Australia and Janssen. And we'll be focusing on these three key areas now and into the future. The first is on evidence. Where there are clinical trials available, patient advocacy groups and your physicians and nurses can be a great source of information on what might be relevant to you at a point when it's available. And registry data, the importance of collecting information so that we can see how medicines are performing once they're in market. What is your experience? As I said, that patient experience index, understanding yours and your carers' journeys is critically important, particularly as we move into new therapeutic areas like inherited retinal diseases, and mapping the moments that matter most so that we can seek to work with you to find the appropriate solutions. And lastly, again, definitely not least, is access. Australia is a complex beast when it comes to health technology assessment, what processes people go through, be it PBAC or MSAC or some new process that doesn't currently exist. We will be coming to talk to you because I can tell you we would never have shaped the change that we did in Australian access to medications for a life-threatening condition without the voices of the people who lived with them. And this is a brilliant segue for me because Marija's coming very shortly to talk to you, and that is the importance of genetic testing. And I'm not going to steal her thunder because she knows far more about that than I do. So without further ado, I'd love to say thank you very much. Sally, I believe you said we'll be taking questions at the end, so I won't ask for them now, but please do think of some and I'll be here to answer them after Marija's talk. So thank you so much for your attention, and Marija, looking forward to hearing from you.
Thanks so much, Felicity. I'll just introduce Marija while she gets us off ready. Marija McGeachie is the Senior Medical Advisor with Janssen, and her topic today is inherited retinal diseases, the role of genetic testing and potential future treatments. Marija holds a PhD in medicine from the University of Sydney, and her PhD work focused on identifying genetic causes and molecular characterization of inherited eye disorders. Following completion of her PhD, Marija then undertook a postdoctoral appointment with the UCL Institute of Ophthalmology in London, where in close association with experts from the Moorfields Eye Hospital, she developed gene therapies for inherited retinal dystrophies. Since her return from the UK in 2012, Marija has held numerous roles in medical affairs within the industry, both at Alcon and Johnson & Johnson. Marija is currently the Senior Medical Advisor looking after Janssen's leukemia and lymphoma portfolios, as well as the pipeline ophthalmology portfolio. So welcome to the webinar, Marija, and we'll look forward to hearing from you.
Thank you so much, Sally, for that wonderful introduction, and good afternoon, everyone. So as you heard, my name is Marija McGeachie, and I'm the Medical Advisor at Janssen Australia. I'm very excited to be here today because, as Sally said, ophthalmology is very close to my heart, as I've spent the entirety of my academic career studying ophthalmology. Now, I would like to start today by talking about why Janssen is entering the ophthalmology space. And some of you may know that Johnson & Johnson at the moment, or Janssen rather, doesn't have a portfolio in ophthalmology. And then we'll pivot to talk about inherited retinal diseases more specifically, or IRDs as I'll refer to them from now on, and the innovative therapies that are now really becoming a reality. Now, these innovative therapies are really a hallmark of precision medicine, and as such are tailored to very specific molecular defects. As a result of that, and Felicity mentioned that earlier, diagnosis or genetic or molecular testing will be a critical step in accessing some of those treatments. And we will touch on this at the end of the presentation too. Now, I'm sure that it doesn't come as a surprise to anybody in this virtual room that retinal disorders can have a huge societal impact, and that visual impairment and blindness that are associated with retinal disorders impact more than 350 million people worldwide. And more specifically, IRDs can affect anywhere between 1 in 1,000 to 1 in 4,000 people, depending on which journal article you read, and are the leading causes of blindness in working age adults, and that certainly holds true in Australia. And in Australia, IRDs are actually the second most common cause of blindness. So there is a very, very high unmet need here. And as you know, for a lot of these disorders, we don't really have a treatment to offer to these patients at this point in time. Now, before I start talking about why Janssen has chosen to really invest in this space, I really want to take a minute to reflect on my own academic journey. And I remember when I was in my academic role back in London and working on these gene therapies for IRDs. And I remember very distinctly one day, after having finished a whole heap of assessments, I led all the preclinical work on some of these genetic disorders, and finished a whole heap of assessments on the animal cohorts that we were doing. And I was thinking to myself, which went amazingly well, and I was thinking to myself, well, you know, I'm holding this tool that can be absolutely transformational for some of the patients who are living with these diseases. But it was a very depressing thought, because at the time I thought, you know, because they're very rare and or labeled as very rare diseases, this is never going to be commercially viable. And I can't tell you just how excited I am to be here today to talk about this as a possibility of access for our patients to these therapies. And Janssen is one of the companies who have decided to invest in this space because of that high unmet need. And our vision as a company is to restore and preserve vision in patients with retinal diseases through those transformational therapies. And those transformational therapies will be the innovative technologies some of the innovative technologies that we'll be speaking about today. Now, Janssen is looking at really investing into 5 different therapeutic areas within the retinal disease, all of which have very high unmet need. And they include IRDs, glaucoma, wet AMD, dry AMD, as well as retinopathies associated with diabetes and macular edema. Now, today, we will specifically focus on IRDs, and more specifically on X-linked retinitis pigmentosa, or Xlrp, as I'll refer to from now on, because this is one of the treatments that Janssen is partnering on with a company called Marija in the UK on a gene therapy treatment for Xlrp. Now, as we all know, Xlrp is a serious and progressive IRD that causes significant visual disability. And Xlrp is a subset of retinitis pigmentosa, or a pool of diseases that cause loss of visual function. And there are currently around 250 different genes that are known to cause IRDs. And there are many more genes that haven't been identified as yet. And the way each IRD is passed from generation to generation can be different in families. You can have autosomal dominant inheritance, autosomal recessive inheritance, or X-linked, for example, which is what we're dealing with here. And X-linked simply means that that genetic defect is located on the X chromosome. As I'm sure you all know, males have 1 X chromosome, females have 2 X chromosomes. And as a result of that, because males don't have that second backup of the second X chromosome, so they're the ones who are mostly experiencing Xlrp. However, there are females who are also symptomatic. Now, majority of Xlrp cases, between 70 and 80%, are actually caused by a mutation in a gene called retinitis pigmentosa GTPase regulator gene, which is a bit of a mouthful, so I'll refer to it as RPGR gene. And what does that gene do? So as you know, genes are blueprints for proteins, and proteins are the doers in our body. They're the molecules that actually provide the function. And at this point in time, we don't actually have a very clear idea of what RPGR actually does. However, there is pretty good data to provide a good hypothesis as to what its function is. And what I have on this slide here is a beautiful schematic representation of a photoreceptor cell, specifically a rod. As you know, we have rods and cones that help us see. And those are highly specialized cells at the back of the retina. And they have inner and outer segments. And inner and outer segments are connected by what we call a connecting cilium. Now, RPGR plays a role in the function in the ciliary gate, in that cilium, which controls access for various molecules that are being transported from inner to outer segment. So it's very, very critical for the actual function of the photoreceptor cell. So what are some of the core symptoms of Xlrp? Children and early adolescents, some of the first symptoms that they will experience is night blindness. And if you have children of that age, which I certainly do, I know that that can be quite frightening for them. From there on, they will be experiencing a decrease in their peripheral vision and start to experience tunnel vision. So they'll be able to see through that sort of central button, which will slowly be closing in on itself until it's completely extinguished by about third or fourth decade of their life, when they're going to be completely or legally blind. Now, having said that, you can see that the prognosis for these patients is very, very poor, and particularly because there are really no treatment options to offer these patients at this point in time. But their journey is also often quite complex, and just going back to what Felicity was talking about, that patient journey. What I've got here on this slide is a patient journey that was mapped out by our US team of what happens normally in the US, but it's not really that different anywhere else or in Australia. So you've got a patient that's identified either through school or at home, and they might circle around that primary care or early healthcare professionals who may or may not have awareness of Xlrp or the ability to diagnose before they actually get to the IRD specialists who can provide a proper clinical diagnosis, which is a bit of a milestone. Now, those patients then may or may not go through genetic screening to determine the genetic diagnosis. And if you think about it from the perspective of treatment availability, that makes sense because there's no treatment available. So why would I get tested to understand my genetic diagnosis? But, and I will come back to it at the end of my presentation, because there are actually reasons why it's very important to know what your genetic diagnosis is. And those patients will generally then go through a preparation process for living with a decreasing vision and eventually blindness, get connected to support groups like yourselves, and then being maintained as Felicity was speaking about earlier. Now, why is this important to us, and identifying all of these barriers that you might see as red points on this slide? Again, going back to something that Felicity was saying earlier, for us, it's not just about providing that treatment, it's about that entire experience. Because understanding your journey is not the same as understanding your experience. So it is really, really important for us to understand those moments that matter to you and those points in the journey that are barriers and pain points, so that we can really meet that need, work together with your clinicians and support groups to meet your needs, individual needs. Now, I'll talk a little bit about gene therapy for IRDs in general before before I give you a little bit of an overview of some of the data for the Xlrp therapy. And I want to start by looking at the eye as the target organ for gene therapy. Why would it be a good target organ for gene therapy? And on the slide here, I've got a schematic representation of the anatomy of the eye, which I think is an absolutely perfect structure and a perfect organ. Now, the eye has a combination of features that really make it ideally suited as an organ for gene therapy. And the first one is that it is highly compartmentalized, which really facilitates accurate delivery of the therapy to the tissues that you want to target. And as a result of that, that can minimize any systemic dissemination of the therapy and any unwanted systemic side effects. Intraocular tissues also have small but very stable cell populations. And what that means is that they don't replicate. So a bit like cells of our within our skin, we lose skin cells all the time and we get new ones, etc. That's not the case with the retina. Those cells don't change. What you get as you develop, that's what you generally have for the rest of your life. So that's a very good thing for gene therapy, because it means that we can deliver functional genetic material to those cells and it'll stay there for the rest of its life. So it also provides an opportunity to have once in a lifetime treatment. Now, the other feature is the unique immune environment of the eye. And it's really because of those anatomical barriers. It provides a degree of protection from the immune responses from the rest of your body that can be directed against that therapy that you deliver in the eye. So that is good, because otherwise, if your immune system is able to target that therapy, it can cause inflammatory processes, and inflammation is not something that we want to see with these therapies, because it can actually affect the therapy itself, and it can actually decrease the level of efficacy of that therapy. And we will come back to that a little bit later when I talk through some of the data. So it is a very important point. The eye also has a very unique optical transparency, which enables non-invasive ways of imaging ocular tissues live. And we can very readily quantify visual function, which is very good. Now, the final thing that I'll mention about the eyes is one that not many people consider as a good feature, but the fact that we have two eyes is actually a very good thing from an experimental perspective. And you will see again later on when I talk about some of our results, in that when you have a systemic therapy that you deliver in a clinical trial, you have to choose a patient who satisfies the criteria that you're looking for. You then have to find a patient who's pretty much exactly the same to act as a control, because you want to be able to compare apples to apples at the end of that trial when it comes to the outcomes of that therapy. And so with the eye, because we have two of them, we can have one treated and one untreated, and provides an internal control, which is actually it's invaluable. So let's talk about how gene therapy works. And I'm sure a lot of you are already familiar with this. But what are we actually trying to do? Is this a curative treatment? And I think it's really important to set expectations in that these therapies that we have today, with the technologies that we're able to deliver, are not going to be curative, in that the patient is able to reverse the disease progression to the point that they were before they had any symptoms. So we're only targeting, part of the reason for that is because we're only targeting a certain area of the retina around the macula, and we'll talk about that in a minute as well. So what we're trying to really do here is arrest the disease progression, preserve any remaining vision, and restore vision as much as we can around that targeted area. And really what we want to do is offer better quality of life for those patients so that they can feel confident in their day-to-day activities and they have that functionality. Now, how does it actually work? So we're starting with that aberrant gene that has a mutation. In this case, we're talking about RPGR. The first thing that we need to do is engineer a functional copy of that gene. And a functional copy of that gene will be packaged into a vehicle. And we have to have a vehicle to take that genetic material into the nucleus of the cell that we are targeting. And the vehicle that we're using in this case is attenuated virus, and we're using adeno-associated viruses. And that was a very, very clever scientific find because AAVs have the ability to be very specific for some tissues and cells. So we can be very, very targeted about which cells we're going to deliver the material to. So once packaged into that vehicle, it is delivered as a suspension, as a subretinal injection. It's a surgical procedure. So what I've got on the slide here is an insert that really shows those beautiful layers of the retina and shows where that injection is actually is going to be delivered. And that's between the photoreceptor layer of the retina and the retinal pigmented epithelium. So it is quite a delicate procedure. But we're very, very fortunate in Australia in that we have internationally recognised clinicians who have been trained in these, in delivering these therapies. Now, I'm going to show a bit of an animation around what happens once the virus actually gets in there, in the eye, and I will talk it through as it's evolving. So with subretinal injections, we get a bit of a bleb, so a little bit of a retinal detachment at that area, which resolves pretty quickly as the suspension gets absorbed into the cells. Once in the cells, that genetic material from the virus finds its way to the cell nucleus, where the rest of your genetic material lies. And what it does then, it actually just sits there and uses the cell machinery to produce a functional copy of the protein. And because, as I said earlier, those are very stable cells, they're not replicating, you're not losing that genetic material. It's theoretically there for the rest of your life. Now, where do we go from here? Well, as I said earlier, I think we're very, very fortunate in that we have internationally recognised expertise in Australia. And not only that, I believe we're one of two countries in the world who have guidelines for the assessment and management of patients with IRDs. That sits under the umbrella of the Royal Australian New Zealand College of Ophthalmologists. And the guidelines recommend that the assessment and management of patients with IRDs falls into 4 areas. The first one is establishing that clinical diagnosis. The second one is determining the level of visual function and then implementing any visual rehabilitation that's needed. The third one is establishing genetic diagnoses and genetic management. And the fourth one is around monitoring the disease progression and then subsequently planning for any therapeutic interventions if available. Now, 1, 2, and 4 are pretty much standard of care, but what I really want to focus on today is the third one, which is establishing the genetic diagnosis. And the guidelines really provide a very good rationale as to why establishing a genetic diagnosis is important. And that's to help provide a more accurate diagnosis and prognosis. And I highlight that because somebody who has a mutation in one part of the gene versus some may not have the same prognosis versus somebody who has a mutation in another part of the same gene. So we're getting very tailored here to those molecular defects. So it's very, very important to know what your genetic diagnosis is. It will also provide individuals and families with specific recurrence risks, will inform their reproductive decisions as well as treatment decisions down the track. And depending on the mode of inheritance, testing may be important for other family members as well. So for example, if your disease is autosomal recessive disease, it means that you need 2 defective copies of the gene. You will have 2 defective copies of the gene, and you really need to confirm that, because any treatment and trials will have that as their inclusion criteria. If you have autosomal dominant disease, it means you have 1 defective copy of the gene. It's important to examine parents and siblings to determine whether they're presenting with different levels of symptoms, and also to identify those people who are also at risk. With X-linked disease that we talked about earlier, there may be asymptomatic carrier females, for example, and other family members who may benefit from genetic testing. And as I mentioned earlier, understanding the precise molecular defect is really critical for implementation of any genomic therapies. Now, it's important to note that despite the advances in technology over the last couple of decades, a genetic answer may not be found in up to 20 to 40% of cases. And that's because we still don't know, we still haven't identified all the genes that are responsible. And this is the reason why the testing should really be performed in collaboration with your clinician and a genetic counsellor. And you might have genetic testing performed as part of your clinical diagnosis or as part of research, but it should all be delivered with appropriate pre and post genetic counselling. Now, I want to emphasise here also that genetic testing is not formally reimbursed in Australia. And as Felicity has mentioned earlier, there are a lot of regulations around genetic testing in association with a delivery of treatment. And so for those therapies that are not yet available, we can't really tell what those regulatory requirements will be. Therefore, it's important to note that if you have been tested so far, should a treatment become available in the future, you may or may not need to get tested again to satisfy those regulatory requirements for that treatment. So I just wanted to make a couple of those points around genetic testing. Now, I'd like to just finish up by going back to what Felicity opened up with, in that really emphasizing that our commitment is to help bring people with retinal diseases a brighter future. And Felicity talked about engagement and innovation, and I talked about the innovative therapies that we are still developing, and the importance of genetic diagnosis. However, as Felicity said, that engagement is going to be critical for us in the coming years, and we'll be working with your clinicians and Retinal Australia as well to really find ways to really understand your journey and incorporate your needs within any therapies that are coming down the track. So with that, I'll close, and I'd be happy to take any questions.
Thank you so much, Marija. We do have time for some questions now, and Felicity is here as well, so we can have everybody. So if you want to ask a question, just type, you can type something into the Q&A section, or you can use the raise hand option at the bottom of your screen, and we can respond to those in the order that they appear, and we can help you to turn your microphone on so that you can ask a question. So if you'd like to do that, you can also use Alt-Y on the keyboard to raise your hand. Here's a question from Heather Mack. Go ahead, Heather.
Thank you. That was a very interesting presentation. Can you please tell us what other genetic therapies are in the Janssen pipeline for retinal diseases?
So The one other one that I can share with you is achromatopsia. So we've got trials that are starting for CNGA3 and CNGB3 genetic defects. We are working with our local clinicians actually to see whether there is an opportunity to have that trial in Australia as well. Others are a little bit you know, further down the track, so I won't be able to talk about them today.
Thank you.
So now we've also got a question here from Mark Wong, and he wants to know how many stages are there in the clinical trial?
So in this particular clinical trial, there were 3 stages. So there were 3 phases that we had in phase 1 and 2. And actually the last phase of that trial was actually the foundation for the phase 3, which is at the moment is recruiting in the US and the UK. So we're now at the stage where we're recruiting for phase 3, for which results we'll hopefully see over the next couple of years.
I'd like to follow up on that question myself. I was interested to know that obviously it's in the pipeline now. What would be the timeline, assuming that the trials go well? When would people be expecting to see this product on the market?
Yeah, that's a very good question, Sally.
I'm sorry, question everybody asks.
Not one easy to answer, because if everything goes well, we still have to, and as Felicity was saying earlier, there are a lot of regulatory requirements that we have to meet. So it's a lengthy process to try and get it into the market. So it's still like a few years away until we see the results, and then that will kickstart all those processes if the results are favourable. So sorry, I can't provide you with the exact timing, because really, you know, a lot of it is not within our control, if that makes sense.
I understand, but it is terrifically exciting to see how well it is, and fantastic to get a little bit of an understanding of the patient experience and being part of a clinical trial and being able to see exactly how you test the efficacy of the treatment is fantastic. Does anyone else have another question? Feel free. I have another one. I can keep going. I've written a few down actually. It may be sort of an obvious one, but just going back to the way you administer the drugs. I'm assuming that when you use the adenovirus, you're actually sort of infecting the cells, that you don't have to get it into all the cells because the virus will infect all of that layer of cells. Is that correct?
Yeah, that's correct. It's a really clever technology actually, because as I said, we're using viruses that have the ability to target specific cell types. So we specifically in this therapy, we're specifically targeting photoreceptors. And the way that we genetically engineer that material that we package into the virus also helps it being that gene being expressed in the right cells. So we're really being as targeted as we possibly can.
Fantastic. And I've just got one other question. You mentioned that in the initial stages you had males being tested. And I'm interested, I guess, the two parts to the question. Is there a reason why you chose just to do men at the start? And is there, like other medical testing where it has become clear that actually it's important to consider male and female responses to drugs? Is there a known difference between eyes, you know, and the way eyes react in males and females?
Yeah, that's a really good question, Sally. So initially they've chosen to only recruit males just because it's X-linked inheritance. So it's males who are generally affected with Xlrp. So as I said, males have 1 X chromosome, we have 2 X chromosomes. So we sort of have a backup gene there that if functional, you shouldn't have any symptoms. However, there are some females who do have symptoms, and I think that was recognised through this process, and we're now recruiting both genders. In terms of whether there's any difference anticipated in reaction to the therapy, there isn't anything that we're aware of that there is a difference between genders.
Great. We also have a question here from Chris Gleeson. So I'll just allow you to talk, Chris.
Thank you and good afternoon, and thank you all of you for what you're doing. My question is, after listening, that these trials seem to be happening overseas. I'm just wondering whether or not there is access to similar trials here in Australia.
Yeah, that's an excellent question, Chris. And we are we're very big advocates of getting the trials here into Australia. So myself and my colleagues at Janssen here locally have been working very, very hard to attract those trials into Australia. And as I said, because we have such amazing expertise in Australia that we're already starting to work with, I do believe that there will be an opportunity for our patients to be involved in some of the trials that are coming through. This particular trial, unfortunately, was started, I think, even before Janssen got involved. So, you know, the protocol was set, the sites were chosen, so there was no opportunity. But certainly for any of the other trials that are coming through our pipeline, and not just with IRDs, but in other disease areas that I've talked about, we will be absolutely advocating that Australia be a part of it. And we will certainly, you know, make that known through our clinicians and Retinal Australia as well.
Thank you.
And we have one final question here from Mark Wong. Is the virus injected directly into the eye, or is it just in muscle tissue such as your arm?
Yeah, no, good question. So it's directly injected into the eye at the back, so into the eye and to the back of your retina. However, in contrast to some of the other therapies that you might have seen for some of the other retinal diseases, this will be once in a lifetime injection. So once the virus is delivered, that genetic material should theoretically stay in those cells for the rest of your life.
Fantastic. Okay, I'm just going to ask one last question, and I wanted to ask Felicity actually, because you were talking about you wanting to get feedback from patients about what is going to work best for them and how those different things. How do you access those people? I mean, would that be during a clinical trial, or are there other ways that you access members of the public who you want to find out information about?
So pleased you asked that, Sally, because patient advocacy groups are a absolutely key conduit to being able to have people raise their voices. We live in a very regulated environment in Australia from a healthcare perspective, and so we have a protocol. We use an independent research agency, and oftentimes we will introduce them with the shall we say, the partnership of a patient advocacy group such as Retina Australia or Leukemia Foundation, Lung Foundation Australia, for example, just to give you an idea of some of the groups we've worked with already. And then they are able to communicate to their members that this research is available. If people would like to participate, they speak independently to the research organisation, and what we get back at the end of it is a de-identified report with people's feedback that they are willing to share, mapped out against those zones that I was showing you across the patient engagement index. And that gives us two great pieces of information. One is that we can overlay it. If you look across the breadth of Janssen's portfolios and everything that we work on, from oncology to immunological conditions, pulmonary arterial hypertension, the ophtha portfolio moving forward, the neuroscience diseases, for example, and lay those experiences over the top of each other, you may be surprised how common some of the problems are amongst groups, because, hey, we're all people and we all have similar problems when we're met with complex healthcare needs, particularly in that need for care and support and education and information. And then there are what one of my lovely colleagues refers to as the unicorns, those things that are a moment that matters in a particular area, a particular disease state, sometimes at a particular point in the journey. And it's imperative that we uncover those too, because we can meet the needs of most of the people most of the time, but we also want to meet those unique individual needs in particular disease areas. So the research is incredibly valuable from that perspective. So when we're able to do it, any of your members who are interested to share their stories, we would be thrilled to hear them.
Fantastic. Thank you so much. All right. Well, thank you very much, everybody. I'd like to just invite Leighton Boyd to join us again to say a final thank you.
Thank you, Sally. On behalf of the board and all attendees at today's webinar, I'd like to thank Felicity and Marija for their interesting and insightful presentation. Janssen has been a strong supporter of Retina Australia, and I look forward to continuing that relationship. We are also excited to hear that Janssen is moving into research into inherited retinal eye diseases. As the largest patient group in Australia, we are always excited to hear of research that will hopefully benefit us into the future. Thanks again for today. Thank you, Sally.
Well, I hope that you found the session informative this afternoon. We've certainly been delighted to present it to you, and it's just fantastic to hear about the work that Janssen is doing. If you have any more questions, don't hesitate to contact us. You can just email us in the office and we will try and assist you or put you back in touch with one of the presenters if they can provide more information. So that's the end of the webinar, and thank you for joining us. Thank you again to Felicity and Marija for your time, and thank you all and enjoy the rest of your day.
Thank you so much.